So far, we have learned the proteins and carbohydrates. Both are one of three energy sources. The last one is fat or lipids. And in this lecture and the next, we'll discuss lipids and their types and structures, as well as biomembranes and membrane transport. Lipids are a class of biomolecules with a hydrophobic character, meaning that they have a poor water solubility, and they play a large role in energy metabolism, as well as energy storage as a form of fat, and are major structural components of biological membranes. And an example of lipids is a phospholipid, where you can see its structure here, and that has the amphiphilic property. And you should know what that means, of course, that has both hydrophilic and hydrophobic residues within the molecule. And that property helps to form the biological membranes. Again, lipids have a structural role to form the membranes. And these membranes serve to organize biological processes, which occur in distinct compartments within the cell. And also, membranes are a component of cytoplasm separated from organelles, as well as the exoplasmic compartment. And the membranes are not totally impermeable, so they're not like a Ziploc plastic bag that doesn't allow any water or anything go through once it's zipped up. But the membranes have regulated flows or exchanges of molecules via gates and pumps. And also, diffusion works for some molecules. And lipids have other functions as energy storage. An example is adipocytes, or the fat cells that basically deposit the fats. And also, they're involved in signaling functions. Examples are steroid hormones, prostaglandins, glycerol, and they're involved in many different signaling pathways, such as kinase signaling pathways, and so on. Again, lipids are biological molecules that are insoluble in water and soluble in nonpolar solvents. And for the classification of fatty acids, there are different classes here are listed. And we're going to go over one by one in this lecture to study more about their structures and their properties and their functions and so on. So the first one listed here is the fatty acids, which you can think the form its structure as just a half of this structure written here that contain a polar carboxyl group as a head and a nonpolar hydrocarbon chain as a tail. And another class is triacylglycerols, which you can see here that the three fatty acids are basically linked together, and they are the storage forms of fatty acids, so are going to be used as an energy storage function. And another one is glycerophospholipids, which is basically a lipid that contain the phosphate and that have the principal component of the biological membranes. And the next one is sphingolipids, a class of lipids containing sphingosine bases. An example would be the sphingosine ceramide, sphingomyelin, cerebroside, ganglioside. All contain an 18-carbon amino alkyl backbone, and also they are components of a biological membranes. So these are really important, so we're going to revisit them in this lecture. And the waxes, which you can see its structure here, the waxes are the esters of long-chain alcohols with the long-chain fatty acids. And terpenes are the isoprene units, and steroids are another quite important lipids that have characteristic fused ring structure. And a well-known example is a cholesterol, which is the most common steroid. It is a precursor of many important biomolecules such as hormones. So among the different classes of lipids, the first one is fatty acids, which contain carboxylic acid group or head attached to a long hydrocarbon side chain or tail. So the fatty acids have a polar heads and the non-polar tails. And the carboxylic group can be ionized at physiological pH, and the length of tail ranges from 14 to 24 carbons. And most common, the length will be the C16 and C18, and sometimes they can have these double bonds in the tails to make the unsaturated form, as opposed to the tails with the all single bonds between the carbon atoms will be the saturated fatty acid. Here are some examples of fatty acids, especially ones that have 18 carbon atoms. And number 1 and number 18 are indicating the carbon numbers. So the numbering of the carbon atoms starts from head to tail. So start from carboxylic acid to the end of the tail. And all these 4 different types of fatty acids, stearic acid, oleic acid, linoleic acid, linolenic acids, are basically having all the same number of carbon atoms, but have a different number of double bondings, or the slightly different in terms of their structures, and that definitely changes their properties. So we're going to take a look at each of the structures as well as how those structures affect their properties. The first one that we're looking at is the stearic acid, or the stearate, which melting temperature is 69.6 degrees C. And shorthand notation for that will be 18-0, where 18 means the total number of carbon atoms, and 0 means the total number of double bonds. So obviously there's no double bonds, so it's going to be 18-0. And an example for such shorthand notation will be palmitic acid, which is 16-02. The second one here is an oleic acid, which obviously has one double bonding between carbon 9 and carbon 10. And notably, its melting temperature is 13.4 degrees C, which is considerably lower than the melting temperature for stearic acid. And the oleic acid's shorthand notation will be 18-1. Again, 18 is total number of carbon, 1 is for the number of double bonds, which is here. So it's happening at the carbon 9, so there's a 9 indication here. Especially that happens actually between the atom 9 and atom 10. And this double bond, or the existence of double bond in the fatty acids, mean that this fatty acid is unsaturated, and this double bonds usually happen to cis, so they're in the same side. As you can see here, multiple double bonds are typically on the same side of the molecule, and also usually happens at every third bond. So 9, and the next one is 12, and the next one will be 15, and so on. And so in polyunsaturated fatty acid, you're going to see a lot of double bonds, but again follows mostly these 2 rows. But almost never they are conjugated, meaning that single bond, double bond, single bond, and double bond kind of form. It's going to be usually at every third bonds. The third one, the third fatty acid that we're looking at here is linoleic acid, and melting temperature now is -9 degrees C. Again, much lower than both stearic acid or linoleic acid, and it is because of those number of double bonds, which you have 2 here between 9 and 10 and 12 and 13. And the last one is linolenic acid, which melting temperature is even lower, and it has 3 double bonds, 9, 10, 12, 13, and 15, and 16. And this table 9-1 shows the common biological fatty acids that you need to get familiar with, especially their shorthand notation. Again, the first number would mean the total number of carbon atoms, and the second one after colon means the number of double bonds. And the palmitic acid, stearic acid, and so on, you should get familiar with their structures too. And when you look at the melting temperature, actually, there have a correlation between the length of the tail and the melting temperature. So as the fatty acids gets longer and longer, they're gonna have the higher melting temperature, which actually makes sense because they're gonna be more of the nonpolar relation, or the interaction with the other the fatty acid molecules. And also unsaturated fatty acids are listed here, such as oleic acid, linoleic acids, and so on. And again, interestingly, there is anti-correlation between the number of the double bonds as well as the melting temperature. So as the fatty acids have more and more unsaturated, meaning that they have more and more double bonds, they're going to have lower and lower the melting temperature. So here's the recap of what I just talked in the previous slide, as well as a little bit more information. Single CC bonds, carbon-carbon bonds, are almost free to rotate and very flexible. So it can be, it can have any kinds of conformation there, but the most extended conformation, which resembles sort of the line rather than some kind of flap, is most stable because it has minimal steric interference. And when you look at the melting points of fatty acids, for saturated fatty acids, which does not have any double bonds, the melting temperature increases with the molecular mass or the length of tail, and the long chains can pack closely together in extended form, so they're going to have strong hydrophobic interactions. So the shorter ones, saturated fatty acids are going to be oils, but the longer ones are going to be solid fats. And again, think about the melting temperature and its relationship with the molecular mass. And for unsaturated fatty acids, the melting temperature is lower than corresponding saturated fatty acid because they do not pack as well, and double bonds within this tail introduce kinks about 30 degrees, so it will have a rigid band there. And their structure of the unsaturated fatty acids we are going to discuss later on in this lecture. And soaps are basically salts of the fatty acids, and fatty acids rarely found free in nature, but usually they're esterified to something to be stabilized. Now let's take a look at other classes of lipids, which are triacylglycerols and glycerophospholipids, as well as sphingolipids. They all have a similar structure and function too, where it has glycerol as head or the sphingosine as head, and multiple fatty acids are attached to them to form the tails. So they basically have a polar head and nonpolar tails, which can form the lipid bilayer in aqueous condition. And this property confers a very important function for the biomembranes. First one, the triacylglycerols, which structure is based on glycerol, but instead of having these hydroxyl groups, these carbon atoms are attached to acyl groups, but there are 3 acyl groups to form the triacylglycerol. So the triacylglycerols are basically triglycerides of neutral fats, and they are nonpolar and water-insoluble fatty acid triesters of glycerol, and they can have simple or mixed triacylglycerols based on the type of fatty acids that are esterified. So if that is just one simple kind for all these 3, that's going to be simple triacylglycerol, as opposed to more than one type will form the mixed triacylglycerol. Here's an example of a mixed triacylglycerol, which is with the palmitic acid, linoleic acid, as well as the stearic acid. So its name would be 1 palmitoyl oil, 2 linoleic oil, 3 stearyl oil, glycerol. Triacylglycerols function as efficient energy reservoirs in animals, as the one molecule contains 3 fatty acids there. And triacylglycerol yield more energy upon breakdown than sugars do, more than twice much, as 9 kilocalories per gram that you can get from the triacylglycerol, as opposed to 4 kilocalories per gram for the sugars. Furthermore, it's not water saturated, meaning that water molecules are not involved in compaction, so more compact, less room needed to store for the triacylglycerol. And taken together, fats provide 6 times more metabolic energy than equal weight of glycogen, so fat reserves allow to survive starvation for up to 3 months because of so much of energy they can store and generate. And the fat cells, or adipocytes, are specialized for synthesis and storage of the triacylglycerols. And also, they serve as insulation layer in whales and Arctic animals. Triacylglycerols are most abundant class of lipids in the cell or the living organisms, but they are not usually found in the biological membranes, but are mostly used in the adipocytes for the energy reservoirs. The next one is glycerophospholipids, also known as phosphorylglycerol, glycerol phosphatides, or phosphoglycerides. They are the subclasses of the phospholipids. As you can see in their structure, they're based on the glycerol again, but at the third position, third carbon here, the phosphate group is attached. So that's why it makes it as the phospholipid. And they are the major components of the biomembranes. And when you look at the structure of the glycerophospholipids, the fatty acids are esterified at position 1 and position 2 here. But at position 3, there is a phosphate group. And the simplest glycerophospholipid would be a phosphatidic acid, where this X is basically hydrogen atom. And the phosphoryl group is not esterified to the other functional groups. So, but there's only minor amount exist in the biomembranes. But usually, instead of just a hydrogen, this part is esterified, and based on the nature of this X group, that changes the property of these glycerophospholipids. And in glycerophospholipids, the phosphate group that is attached with the other functional group serve as a head group, as opposed to the fatty acids that are esterified into the other positions of the glycerol backbone would be the tails. So based on the different type of this functional group that are attached to the phosphate group, they have different identities or the classes of that glycerophosphate. The first one here is the phosphatidylcholine. Obviously, choline is attached to the phosphate group. And also there are some more examples here. Here you can see the phosphatidylethanolamin. Ethanolamine is attached there. Phosphatidylserine, of course, serine is attached to the phosphate group. And the glycerol can be attached there to form the phosphatidylglycerol and so on. And also, it forms this glycerol form again to form the diphosphatidylglycerol too. This table shows the common classes of glycerophospholipids and lists the X group here that can be attached to the phosphate group. So it can be again hydrogen atom or the ethanolamine, choline, serine, inositol, and so on. And among them, phosphatidylcholine and phosphatidylethanolamine are two most common constituents of biomembranes. And all of these are amphiphilic or amphipathic, meaning that they have polar heads and nonpolar tails, and they have both the properties of hydrophilic and hydrophobicity too. And these are spontaneously making the micelles or the bilayers that we already talked about their properties and their shapes in the chapter 2 about the water. Phospholipases are the enzymes that break down or digest the glycerophospholipids molecules, and they have different classes. For example, phospholipase A1 attacks specifically on the ester bond of the position 1 that breaks down this region to release the R1 group or the fatty acid at the position 1. As opposed to A2 attacks to the second position, and phospholipases C or D basically breaks down this phosphate group to release this X, the functional group. And after the phospholipase action, then phospholipids will become lysophospholipid. And another lipids that have a similar structure, which is based on the glycerol backbone, will be sphingolipids, which are also major components of the biomembranes. and they are derived from sphingosine, which have 18 carbon amphiphilic or amphiphilic amino alcohol. And interestingly, it has this amine or the amino group attached at the C2 position 2 of the glycerol backbone. And derivatives are the ceramides. An example is shown here. And the fatty acids are attached via amino group here in the middle. Examples of sphingolipids are sphingomyelin, cerebroside, as well as gangliosides. All of them are ceramide derivatives. And for sphingomyelins, instead of a hydroxyl group, it has phosphocholine or the phosphoethanolamine. And for the cerebroside, it has the glucose or the galactose, which are basically sugar unit, but it has to be single sugar residue. And for the gangliosides, instead of the single sugar in the cerebroside, it has oligosaccharides, including at least one cerealic acid residues. Now let's talk more about these ceramide derivatives. First one is sphingomyelin, which is basically ceramide plus phospho compounds attached to the end of hydroxyl groups, such as phosphocholine or the phosphoethanolamine. And they are the components of a myelin sheath of nerve cell axons, so it has a critical role in transmitting the electricity through the nerve cell by the insulating the electrical impulses. So since it is have a critical role in there for the nerve cell function, myelin sheath breakdown basically occurs in multiple sclerosis patients. The other subgroups are glycosphingolipids, which are ceramides with the sugars attached to their hydroxyl groups. And examples are cerebrosides, gangliosides, as well as serpentides, and so on. All of them are critical components of membranes in nerve cells for their functions. And cerebrosides, again, like I told you in the a few slides back, are the ceramides with a single sugar as head group, as opposed to gangliosides are the containing oligosaccharides as their head group, as well as the sialic acid, which have negative charge too. And the sugar head groups in the glycosphingolipids have several important roles, which are extending outside to the cell surface. So it can serve as receptors for the pituitary hormones, or the determinants of cell recognition too, by having a peculiar functional groups there sticking out of the cell, and also can be work as the receptor for the bacterial toxins such as cholera. And this shows the again structure of cerebroside, and importantly for the hydroxyl group, it has glucose or galactose, basically the sugar units there. And as opposed to cerebroside, gangliosides have oligosaccharides in their head groups. And based on the number of sugars within the head group, they're subcategorized as GM1, GM2, and GM3, where G refers to gangliosides, basically, and M refers to monosialic acid, and the numbering, such as 1, 2, 3 here, is based on its relative mobility in electrophoresis among these gangliosides. So the GM3 is the smallest one here, which contains only the galactose and glucose, as opposed to GM1 is the biggest one, containing 4 different sugars attached to make the oligosaccharides there. And let's coming back all the way up to the different classes of lipids. Let's talk briefly about these two different classes. First one's waxes, which are esters of the alcohol and fatty acid. And it can be used for the waxy coating of the fruits, which is lanolin. And terpenes are another type of lipids, and they're derived from isoprene, which structure is written here. And usually they have intense flavor, color, and odor too, so they can be used as a deterrent or some kind of other fragrance too, such as menthol, limonene, citronella, phytol, as well as beta-carotene too. The last one listed in the classification of lipids is steroids. Now let's talk about their structures and cellular functions. They all share the common structure here, which consists of multiple fused carbon rings. And especially cholesterol is a component of animal membranes and precursor of steroid hormones such as testosterone, estradiol, as well as progesterones. Cholesterol is a component of lipoprotein complexes which serve to transport lipids in the body, which is aqueous condition. So the lipoprotein, which consists of proteins and as well as phospholipids and some other lipids, make the micelle-like structure there and encapsulates the other lipids such as triacylglycerols or the triglycerides. They're the same thing, as well as some other glycerols and fatty acids to the other location. There are 4 different types of lipoproteins. The first ones are chylomicrons, which is least dense lipoprotein, and they transport the dietary triacylglycerols and cholesterols from intestine to liver. And the second one is VLDL, which stands for very low density lipoprotein, and they transport the triacylglycerol from liver to tissues. And LDL, which is a pretty infamous lipoprotein, the low-density lipoprotein, contains 50% of cholesterol and 25% of protein, and it transfers the lipids from liver to tissues, and they are referred to the bad cholesterol. So you want to keep it low in your body, as opposed to high-density lipoprotein, or HDL, is considered as a good cholesterol. So it contains 30% of cholesterol and 33% of protein, and transport lipids from tissues to liver. And good thing about this HDL is it's doing the scavenging of the lipids and transport it to liver for the disposal. And the LDL, especially, like I told you, an infamous lipoprotein, are more susceptible to oxidative damage, and the damaged LDLs are deposited in the arteries, which can cause the atherosclerosis, which is hardening of arteries. Some types of lipids have a critical structural role in forming biological membranes. The biological membranes are so important because they define the inside and outside of the cells. So basically, without membranes, you're going to lose all the goodies inside of the cells, which make it impossible for sustaining life. And the membranes have a critical role in compartmentalization within the cell too. So basically, these membranes define the subcellular organelles such as ER, Golgi, mitochondria, and so on. All of them have a sort of individual, independent organelles, have different biochemical functions happening within those regions. So without it, all the reactions can be just mixed out, or also sometimes you don't want any certain biochemical reactions to happen. So the compartmentalization is critical to be most efficient in the controlling and regulating the biochemical reactions. And also, the membrane surface can be used for certain reactions to occur. So an example would be the electron transfer in respiratory chain, as well as some kind of cell-to-cell interaction or the cell signaling pathway transduction, and so on. These biological membranes are assemblies of lipids, proteins, and carbohydrates. Glycerophospholipids and sphingolipids spontaneously form micelles and bilayers because of their amphipathic or amphiphilic properties, and that is to minimize contact between the hydrophobic lipid chains and the aqueous solvents here and here, which is mostly in the living system. But if we were to be the non-polar condition as a solvent, would have been inside out for the micelles too. And depending on the type of lipids, they can form either micelle, bilayer, or even liposomes. So for the lipids that have single tail like that, which can form the wedge-like shape, it can generally form the micelle because it has a smaller space on the tail side as opposed to head is relatively bigger. But if the lipid single unit looks like a little cylinder here, which can have 2 or even 3 hydrophobic tails, then now the space occupying on at the tip of this tail end and the head group are quite similar. So they're going to actually form this kind of bilayer structure in the water. And also, depending on the shape or the mixture of these two, they can also form this kind of liposome, in which case it can actually form the micelle-like structure. But when you look at it more closely, they're also bilayer too. So inside of this liposome, they can still have aqueous condition here, where they can trap the polar substances or even water too. Here are some more details about micelles and liposomes as well as bilayers. For the micelles, its structures are defined by the critical micelle concentration, or CMC, and the aggregation number N. And general rule is that the longer the tail of its component lipid, it'll have the higher aggregation number N. And with the single-tailed lipids, it'll form the spheres, such as like micelles, as opposed to bi-tailed lipids, such as glycerophospholipids or the sphingolipids, will form the flat bilayers. And for the liposomes, which is a stable structure and can be purified by the gel filtration, the liposomes can make inside and outside compositionally different because it can simply trap some polar or the possibly non-polar component too inside of that liposome. And the liposomes can incorporate channel proteins within those lipid bilayers, and that is to study the transport or the activity of such channel proteins. So make them small, like a cell-like a system there, and then you can test how efficient that is, how it is working, the mechanisms and stuffs like that in vitro. And also, liposomes can be used to deliver drugs and genes too, which is widely used for gene therapy. Membranes are dynamic and fluid-like, meaning that lipid molecules as well as transmembrane proteins can drift about and move here and there pretty rapidly and freely within the membrane. And there are two types of diffusions happening within a lipid bilayer membrane. First one is lateral diffusion, where the lipid molecules can drift about and move here and there within that layer of the lipid bilayer structure. So it happens pretty commonly, as opposed to transverse diffusion is where the lipid molecule flips and moves to the other layer of this bilayer structure. So in that case, it actually requires some enzymes called the flipases or the translocases, which are passive and active respectively. And there are also some random motions for that lateral or transverse diffusions, where the transverse diffusions happens really slowly, but the lateral ones happens pretty quickly. Again, membranes dynamic and fluid-like, so it can have a phase transition too, from gel-like structure to liquid crystal-like structure. So it can be induced by the heat. So before the transition, the membrane, especially the lipid molecules, are aligned really nicely to form the pretty packed lipid bilayer there with the anti-conformation, which is the sort of the extended structure of these hydrocarbon tails of the lipid molecules, as opposed to post-transition, which is more like liquidy, have the gauge conformation where it has generates the kink of the tails, so it will actually make the more spaces there, so it's not as compact as the gel-like structure, which will make this bilayer more flexible and dynamic. So the fluid-like state of a lipid bilayer of a biological membrane is a function of the composition of its lipid molecules and the temperature where it's placed. So phase transition involve changes in organization of fatty acid tails, and depending on their organization as well as components, they exhibit a fairly sharp transition, and at that point of the transition, the temperature is going to be called as melting temperature. So below the melting temperature, the tails of the lipid molecules are packed tightly, so they're going to exhibit the lipid bilayer will exhibit the gel-like structure, and there are going to be little motion of those tails or the lipid molecules, as opposed to that, the above melting temperature, now the lipid bilayer will exhibit the fluid-like state, so the tails are going to be much more mobile. And the biological membranes are in fluid-like structure because they are typically in the above melting temperature condition. And melting temperature is influenced by chain of the lipid tail length. So the longer the length, the higher the melting temperature, and also degree of saturation, so number of single or double bond between the carbon atoms in the hydrocarbon chain. So when the saturation decreases, it will have the lower melting temperature, and also cholesterol content affecting the melting temperature too. So the cholesterol is the rigid ring system. As you can imagine, there are fused 4 rings, carbon rings there. So it decreases the fluidity of the biological membrane. So that means more cholesterol, then higher the melting temperature. But interestingly, very high amounts of cholesterol abolishes the phase transition completely because the tails of the lipid molecules cannot get close enough to achieve gel-like state anymore. This is a cross-section of a biological membrane where you see the inner leaflet and outer leaflet of a lipid bilayer structure. And as you can see here with the different labeling, it has an asymmetry in terms of composition of the lipid molecules between these inner and outer leaflets. Even though the total phospholipids are the same as 50%, when you look at each components there, such as phosphatidylcholine, phosphatidylserine, and things like that, they keep this kind of asymmetry or the differences there, and that is possibly for the efficient regulation of the interaction between the membrane and the cytoplasm or the membrane outer membrane regions. Singer and Nicholson proposed the fluid mosaic model in 1972, which captures a lot of important critical concepts about interaction between the transmembrane protein as well as lipid interacting proteins and the biological membrane. Where they suggested there are intrinsic proteins or the integral proteins that have transmembrane segments and interact strongly with the lipid bilayers. And also there are peripheral proteins or extrinsic proteins that are loosely associated with the biological membrane and also easily removed from that because their interactions with this surface of the membrane is through the electrostatic and hydrogen bonding. And the proteins are in a very dynamic state, so they're going to be constant lateral diffusions and movement, which is rather fast, like a few microns per minute or so. For example, the phospholipid molecule cruises from the one end of a bacterial cell to the other in less than 1 second. So it definitely shows the that dynamic fluid-like state. And the molecules, how they are dynamically moving here and there, the lipids as well as protein along the plasma membrane or the biological membrane.